Assalamu alaikum students. Before going to our educational topic, please subscribe my channel Muntzir Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education, and those students that are preparing privately can benefit from our channel very effectively. Let's come back to our topic, that is fermentation. Fermentation is related to anaerobic respiration, meaning that fermentation is that process when there is deficient amount of oxygen, or fermentation happens when there is complete absence of oxygen. So fermentation process happens in less amount of oxygen or in the complete absence of oxygen. As fermentation is related to anaerobic respiration, and we know that the respiration which occurs in the complete absence of oxygen is termed as anaerobic respiration. So there are two different scientific views about the origin of fermentation. First we see these views, then we will define the fermentation definition, fermentation types, and also we will talk about those organisms that can carry out the fermentation process. So first we see that views which are about the origin of fermentation. First scientific view about the fermentation process is that it is an evolutionary bypass, because some organisms utilize fermentation process so that they keep glycolytic functioning under anaerobic conditions. Therefore, to keep glycolytic functioning under anaerobic conditions, some organisms use fermentation process to obtain energy. The second scientific view about the origin of fermentation is that fermentation is a biochemical remnant, because it has evolved very early in the history of life. During that time, the Earth's atmosphere consisted of no oxygen. So in the absence of oxygen, or in the presence of very short amount of oxygen, the organisms obtained energy from the fermentation process. So these strong evidences are known about fermentation. These strong evidences tells that the common descent of organisms from primitive cells. In these primitive cells, firstly glycolysis and fermentation process appeared. Both these processes, the glycolysis and the fermentation, still persist. As we know that glycolysis produces hydrogen atoms in the form of nicotinamide adenine dinucleotide, that is also known as NADH2 molecule, which you can also see from this glycolytic pathway, that when oxidized form of nicotinamide adenine dinucleotide here at this point combines with hydrogen ions, then it is converted to its reduced form NADH2 molecule. This NADH2 molecule possesses hydrogen atoms which are then transferred to the organic molecules, and these organic molecules, by the receive of hydrogen atoms, by the taking of hydrogen atoms, are converted to reduced compounds, and then these reduced compounds can be an organic acid like lactic acid, acetic acid, propionic acid, or an alcohol. This alcohol can be in the form of ethanol or butanol. This is the main idea of fermentation process and definition of fermentation process, because during fermentation hydrogen atoms which are received from the glycolysis process are transferred to the are donated to the organic molecules, that becomes reduced compounds, and then these reduced compounds, which can be an organic acid such as lactic acid, acetic acid, propionic acid, and these reduced compounds can also be in the form of alcohol such as ethanol, butanol, and some other alcohols. So during fermentation, we obtain either a acidic product or an alcoholic product. So as fermentation process take hydrogen atoms from the reduced form of NADH2 that is produced during glycolysis, as you can see here in this glycolytic diagram. So we can say that fermentation process regenerates NADine, reproduce, resynthesize NADine. As this NADine is needed, is required to continue to derive glycolytic pathway. And we have known in our previous videos that are prepared for glycolysis process that all the living cells carry out glycolysis process to obtain energy to provide ATP molecules. We can also understand this fermentation definition with the help of an equation which will help us to understand the fermentation main idea, fermentation definition. It is as when pyruvic acid, which is the end product of glycolysis, reacts with NADH, that is also obtained from glycolysis. So when both these pyruvic acid and nicotinamide adenine dinucleotide reduced form NADH reacts chemically with each other, then they produce a reduced compound and NADine, which is an oxidized form. Because when this pyruvic acid takes a hydrogen atom from NADH reduced form, then it is converted to the reduced compound. Because this pyruvic acid accept this hydrogen atom from the NADH and is converted to reduced compound, and by donating this hydrogen atom from this NADH molecule, this NADH molecule is converted to its oxidized form NADine, that is present here, and it is oxidized form. So from this equation we can completely understand the fermentation definition in such a way that the hydrogen atoms that we obtained from the NADH, which is produced during glycolytic pathway, this organic molecule here is pyruvic acid, which obtains a hydrogen atom from the NADH molecule, which is converted to a reduced compound. This reduced compound can be an organic acid such as lactic acid, acetic acid, or propionic acid. But this reduced compound can also be in the form of alcoholic product. This alcoholic product can be in the form of ethanol, butanol, and some others. So in this process, fermentation also regenerates the NADine, which is the oxidized form of NADH that is also required for the functioning of glycolysis process. Students, during fermentation, glucose is not completely broken down, is not completely suppleted because considerable unusable energy still traps in the products, except the two ATP molecules which are formed during glycolytic pathway. More ATP is not generated. So fermentation functions only to regenerate NADine, which is the oxidized form of NADH. Next, we will see the types of fermentation. Fermentation consists of two main types. These types depend upon the type of end product that is obtained during fermentation process. The first form is alcoholic fermentation and the second form is lactic acid fermentation. Next, we will see them one by one. Firstly, we study the alcoholic fermentation. The type of fermentation during which the pyruvate is converted to ethanol is termed as alcoholic fermentation. As we can understand it with the help of this equation, it is the chemical formula of pyruvate. When carbon dioxide is removed out from pyruvate, then this pyruvate is converted to acetaldehyde. This is the chemical formula of acetaldehyde. This acetaldehyde is converted to ethanol, an alcohol, in such a way that two NADH2 molecules which are produced during glycolysis takes part in this reaction. So hydrogen atoms are removed from these two NADH2 molecule and it is converted to 2NADine. So now these hydrogen atoms would be attached to acetaldehyde to form ethanol. Ethanol has the chemical formula of CH3, CH2 and OH. As we know that hydroxyl group is the functional group of an alcohol, and when this alcoholic group is attached with two carbons, then it is an ethanol. So in the alcoholic fermentation, the NADH2 molecule reduces the pyruvate into an alcohol, ethanol. This alcoholic fermentation takes place in some fungi such as yeast and in bacteria. This alcoholic fermentation produces ethanol as a byproduct, and this reaction is controlled by the enzymes which are pyruvate decarboxylase and alcohol dehydrogenase. These are the two important enzymes which take part in alcoholic fermentation. Next, we will study the second type of fermentation, that is lactic acid fermentation. The fermentation type during which lactic acid or lactate is generated as an end product by using pyruvate as a substrate is known as lactic acid fermentation. In other words, we can say that when pyruvate is converted to lactic acid, then this type of fermentation is known as lactic acid fermentation. The reason for this lactic acid fermentation is that some animal cells are present which are deprived of oxygen. So they temporarily carry out lactic acid fermentation. We can also understand lactic acid fermentation with the help of an equation. During this equation, suppose we have a pyruvate molecule. This is the chemical formula of pyruvate. Students, keep this important point in your mind that during lactic acid fermentation, carbon dioxide molecule is not removed out from the pyruvate molecule as we have seen during alcoholic fermentation. As during alcoholic fermentation, we have removed a carbon dioxide molecule from the pyruvate. But in this case of lactic acid fermentation, carbon dioxide molecule is not removed from the pyruvate. In this reaction also two NADH2 molecules participate. Hydrogen atoms are removed from this reduced form of NADH and it is converted to 2NADine. Both of the hydrogen atoms that are obtained from the reduced form of NADH2 would be attached to this pyruvate that is converted to lactic acid. It is also known as lactate. By seeing its chemical formula, we can prove that both of the hydrogen atoms have been attached to this lactate. One hydrogen atom is attached here and the second hydrogen atom is attached here. As animals used NADH2 molecule to reduce the pyruvate. Therefore, NADH donates its electrons to the pyruvate to form lactic acid. This lactic acid fermentation is controlled with the help of an enzyme lactate dehydrogenase. Students, lactic acid fermentation happens in the mammalian muscles and in certain fungi. I hope by studying this fermentation lecture you have completely understand that NADH that is generated during glycolysis donate its hydrogen atoms, transfer its hydrogen atoms to the organic molecule such as pyruvate, which is then converted to the reduced compound. This reduced compound can be an organic acid or an alcohol. Next, we will see the types of fermenting organisms. There are two types of organisms which can carry out fermentation. The first ones are obligative or obligate anaerobic organisms, and the second ones are facultative anaerobic organisms. Now we will understand them one by one. The organisms that can survive, that can live only in the complete absence of molecular oxygen are known as obligative or obligate anaerobic organisms. These obligative anaerobic organisms include certain types of bacteria. As obligative anaerobic bacteria can survive, can live only in the complete absence of molecular oxygen. Its example is Clostridium species. The second type of fermenting organisms are facultative anaerobic organisms. Facultative anaerobic organisms and tissues include certain types of bacteria, yeasties, and animal muscle cells. These bacteria, yeasties, and animal muscle cells can ferment nutrients only in the absence of free oxygen, so that they can produce some ATP, they can generate some ATP in the deficient amount of in the absence of this free oxygen, this molecular oxygen. These organisms, this process also provide NADine for glycolysis. Facultative anaerobic organisms and tissues can produce more efficient energy harvesting when oxygen is present, when oxygen is provided to these bacteria, yeasts, and animal muscle cells. So we can say that facultative anaerobic organisms can ferment nutrients not only in the absence of free oxygen but also in the presence of free oxygen. Students, at the end I will also want to tell you that in the absence of oxygen or in very deficient amount of oxygen, fermentation process takes place from the pyruvate molecule, whereas in the presence of very sufficient amount of oxygen, Krebs cycle takes place. At the end, I will encourage you that take care of yourself, your parents, and your surroundings for the betterment of your country and nation. Thank you for watching this your video.